Hello everyone, this is our system and this system is called in vitro low temperature system. That means that we are in the process of making a system that will create low temperature in a very low cost manner, and we can retain different samples, plant samples, fungus samples and the microbe samples, which normally we place in the refrigerator, but placing a refrigerator, there is some problem that it releases some toxic gases and it contains some harmful chemical that also lethal for the environment, and as well as it consumes much of energy that is also indirectly related to the pollution. So here this is the proposal of the prototype of the whole system. So now I will discuss how this system works. So this is the whole system. This is the lid and this is the base. Now from the beginning, the current that is coming from the main system, that is 220 volt, that is going to the SMPS, that is in the inside of the system, lower part, and from the SMPS the current is transforming into the DC current, and the DC current will transmit the current to the Peltier cooler, and the Peltier cooler is present inside the system, and the cooled system is present on the upper side and the heat portion the lower side, and a fan is present inside the system, and from the fan the hot air is coming out from this vent, this plastic vent, and this vent present in two sides for easy evacuation of the temperature, so that the system will run for a long period of time. And we can see this is the temperature display board, that's the digital controller also. So this have two parts. In the lower part is the greenish part, that the temperature we have determined, that is a fixed temperature, that is the 20 degrees Celsius, that in our will we can maintain the temperature, but this is the temperature, the upper part, this temperature is the actual temperature of the inside system. Though we can see that the actual temperature of the room is 31.2 degrees Celsius, but still as the machine is running near about 5 to 10 minutes, the temperature reduced to 26 degrees Celsius. So now I will discuss another thing, that is the timer. This timer is controlling the timing of the inside light, and we can also control the, if required, we can also control the time of the light of the inside system. So if we open the lid, we can see the light is there, the LED light, to reduce the consumption of the energy. And in the inside system, the system, you can see this is a tray is there, this is the metal tray, and the Peltier cooler is cooling the metal tray, and the metal tray is cooling the water present just on the bottom of the system or tray, and the water is circulating by this motor, so that the temperature is temperature can distribute throughout the system. And we can see the probe, that is the temperature probe, that is dipped into the water, so that we can see how the temperature is changing or not. Another thing, we are putting two fungal mother culture. We can see the mother culture of fungal, one of the biological culture system we are maintaining there, and we can also maintain the microbial culture also and the plant culture also. As we know, plant tissue culture media contain the calyces and the micro plants, so this also can be put inside the system. So as we know, to make a whole tissue culture system, we need a vast system and we need some humidity. The water will evaporate very slightly and it will maintain the humidity of the system, so that no need of external AC and the external whole system. As we all know that the AC which run by the electrical current consumes much of the energy and it can produce the other harmful chemical like CFC that also can harm the environment. And another interesting thing that this light can be controlled in our will. We can totally off the light by the switch, so the light will be off for whole day or whole night. But if we want to do a circadian rhythm or want to study the circadian rhythm of the system, we can put it the proper temperature or proper timings of the light, so that the light will only be on particular daytime but will be off on the night time. As you all know, this all system also to maintain this system, the tissue culture lab, it needs vast amount of skill and needs a large amount of time, and it needs a large space to create this system. So now we will close the lid, and again we are showing the whole system from the four side. So this is, we can see this is a portable system and we can maintain it in a very low cost manner, and this system needs very less amount of current. We can also and another interesting thing that we can maintain it by solar energy also, and if we want we can put the current to the UPS of the normal computer so that it can run through 30 to 60 minutes. It is impossible for the freezer also. So our conclusion is that this is the low cost and low maintenance system for growing of different live specimen in a very convincing manner. Thank you very much.